Hi, our presentation will be on the following on Orthomyxoviridae, and the people who are presented are Aryah, Christian, Juliana, and Oscar. In looking at one specific virus, we will look at the overview of influenza A. This virus is a negative sense single-strand RNA. It is also classified as an envelope virus that has a large range of hosts. Those could be from humans, livestock, marine life, and birds. In looking at its overall casing throughout the world, it has a low prevention rate due to the fact that it has 5 million cases annually, as well as looking at endemic locations would be considered North and South America continents, as well as Southern Asia and European regions. Moving on to our next slide of infection cycle and entry. To transfer its viral material, it will use M2 and M1 channels. The proteins undergo replication in the following NP, PA, PB1 and 2 proteins. In transcription, it will release several different structure proteins that either being H1 through 18 or N1 through 11. In translation, it will make a complementary negative sense RNA from a full-length positive strand, which goes more in depth. This virus is an endotosis virus. It is also unique in the fact that it is an RNA-dependent mechanism. This allows for it to travel into the nucleus, although this is an RNA virus. It has to have a primer dependent. This helps in aiding RNA synthesis with using another enzyme or protein to begin the process.
Hello, my name is Christian, and the next part of the cycle is genome replication. The Orthomyxoviridae family consists of negative sense RNA that is divided up into different segments. Depending on the kind of influenza you have, types A and B will have 8 of these segments, types C and D will have 7 of these segments. Regardless, we have to take this negative sense strand, of course, and turn it into its positive complementary strand to be transcribed or replicated. Both these processes are occurring inside the nucleus. However, there's a lot of research that says there's mechanisms that dictate a switch between the two. One of the leading hypotheses is that during transcription, there's these short viral RNA segments that get coded that interact with RNA-dependent RNA polymerase and tell it when to switch to replication from transcription. Another hypothesis is that RDRP interacts with different proteins that also dictate when to switch. Regardless, we're going to take 80 of these segments for influenza A. We're going to replicate them once we have our complementary strands, and then we're going to export them so we can send them to assembly. One thing that's important to point out is that it replicates without a primer, and this is due to a secondary RNA structure known as the panhandle, and basically the 5' and 3' ends are reverse complements of each other. So there are 3 main requirements for assembly and release. Our first is that all viral and subviral components must make their way to the site of assembly. So our replicated genome, our proteins in the cytosol, and our envelope proteins being made through the ER all have to make their way to the plasma membrane. Our next requirement is that all viral components must interact in a specific way to initiate assembly. So basically everything that needs to be on the outside needs to be there, and everything that needs to be inside needs to be there. And then our last requirement is that all the interaction of these components need to come together and initiate this budding complex so it can be released from the cell to infect other cells. So something important to remember is that hemagglutinin and neuraminidase, which are envelope proteins. Hemagglutinin is for attachment to sialic acid receptors. So neuraminidase has a job of, at the budding complex, to clear the cell surface of the sialic acid receptors. So hemagglutinin does just not reattach itself, and the budding cell can't leave. And this is basically the mechanism behind some antivirals that block neuraminidase activity. So for the pathogenesis of orthomyxoviruses, type A and B influenza target humans. Type A leads to more severe diseases and even to most of the pandemics we've experienced. The next one is that hemagglutinin protein, which is the spike protein that's on the envelope that allows it for attachment. Depending on where it attaches, it dictates the severity of the disease. So if the virus attaches to the upper respiratory tract, it's more mild. And if it attaches to the more lower respiratory tract, we can see more severe influenza leading to pneumonia and stuff like that. Moving on, the neuraminidase protein protein has a lot of roles, but one of the roles that are being researched right now is that when it comes in contact with the receptor, the sialic acid receptor, it actually liquefies the mucus protein in the nasal cavity. So it allows the cilia to basically push the virus and allows it an easier access down into the respiratory tract. And the next and finally is that the RNA polymerase genes basically increase or decrease the efficiency of how good it transcribes, replicates, and allows it to go through the replication cycle. And the more efficient it is, usually means it's more lethal. They did an experiment where they replaced the RNA polymerase genes of a lethal strain of flu with weaker ones, and there was a dramatic decrease in the lethality of that strain of flu.
People can become exposed to the virus through droplets from the cough or sneeze of an infected person at a range of 6 feet. Transmission can also occur through indirect exposure to the infectious particles on surfaces and then touching the eyes, mouth, or nose. In order to diagnose influenza in cases of high infections in a population, a doctor can diagnose infections by using visible signs and symptoms. A doctor can also order tests that detect the virus in the form of a polymerase chain reaction test, or PCR for short. The incubation period for the flu ranges between 1 to 4 days, but usually averages at 2 days. Symptoms of an infected person can be asymptomatic or severe. Symptoms include those in the respiratory system, which consists of coughing, sore throat, and a runny or stuffy nose. Systemic symptoms, which consists of fever, chills, headaches, malaise, and myalgia. And other symptoms, which include vomiting and diarrhea, which are usually common among children. Lingering asthenia can also occur, which can cause a lack of strength or energy in some people, which can last weeks. Risk factors include adults 65 and older, as well as adults with chronic health conditions. High-risk groups are also more susceptible at becoming infected. These high-risk groups include, but are not limited to, pregnant people, young children, people with diabetes, cancer, asthma, and more. It is very important to get a flu vaccine, especially if you are in a high-risk group. This will not only reduce the risk of infection, but also reduce the chances of being hospitalized and even death. Vaccinations are the most common form of controlling the spread of influenza. In some occasions, antivirals can be used to prevent or even treat influenza. The most common antivirals used to treat influenza include oseltamivir, zanamivir, and perimivir. All three antivirals are used to inhibit the enzyme neuraminidase, which is found on the surface of influenza particles that allow the virus to be released from the host cell and infect other cells. Two drugs, rimantadine and amantadine, were once used to treat influenza. However, the virus mutated a resistance to both of these drugs, which caused recommending the drugs as treatments to the flu no longer viable.
These next few slides will discuss the evolution of influenza. An important question to ask is, why do we need flu shots every year? For many viruses, annual vaccinations are not necessary, but such is the case with influenza. The answer to this question lies in the process of evolution. Influenza is a rapidly evolving organism. The flu shot that you might have received last year will not protect you from any new strains that will emerge this year. Influenza possesses unique circumstances and abilities that allow it to acquire genetic changes at an increased rate. The factors that increase this organism's accelerated evolution include a large host population, short generation times, and high mutation rates. As shown on the figure, influenza is a zoonotic virus, meaning that it can infect hosts from different species. This ability allows influenza to increase its host population, in addition to providing ample opportunity for genetic reassortment. Genetic reassortment refers to the mixing of genetic material within a species, resulting in novel genetic combinations. For influenza, this process allows it to adapt and evolve at an accelerated rate. The figure on this slide showcases two strains of influenza swapping genetic information after zoonotic transmission. This genetic reassortment leads to the formation of a novel strain. The process of genetic reassortment is exclusive to segmented RNA viruses and, in the case of influenza, drives the formation of novel strains faster than mutation. Understanding genetic reassortment, as well as the evolutionary process of influenza, is important because it allows us to better protect the population from novel influenza strains.
This concludes our presentation, and I hope you enjoyed learning about the Orthomyxoviridae family.